Thank you very much, Matt and Andrea, for the opportunity to share, kind of give an overview of what we've been doing. First and foremost, I just want to acknowledge my amazing team and can't do any of this without them. You see all their faces there. Thank you so much to the growers and producers that we work with. And I'm very fortunate through PBARC and with HARC, the university, SHAC, and many others. This is kind of a collective presentation for the things that we've been working on. And I thought I'd give kind of just an overview, kind of a big picture before I get into coffee leaf rust of the coffee diseases that we have here in Hawaii. And I'll just briefly go over anthracnose and cercospora spot. Sorry about that. It's moving on its own. Get into coffee leaf rust and what we've been doing. And so to begin, some of the, I guess, more minor in the past, the first one, anthracnose, caused by a variety of Callitotricum species. You'll kind of notice what's typical with some of these diseases, that these spores easily spread by rain and wind, high humidity and warm temperatures, of course, encourage infection and spread. And these are some of the pictures that you can see here to the right. As far as some of the symptoms, necrosis or brown spots really can be found on pretty much any portion of the tree, that the leaves, the branches, the floral buds, the fruits, when they're fully ripe, can be affected by these Callitotricum species. And as far as control, this is kind of a common theme with most of these diseases. Pruning the dead material, removing it from the field. If you can cut down on the density and increase the distances to reduce the humidity levels, definitely control weeds, reduce the shade, and there are chemical treatments available. And the other pretty common one in Hawaii is cercospora spot. It goes by many names: brown eye spot, iron spot, berry blotch. It's caused by the fungus Pseudocercospora cophicola. And again, these warm, humid temperatures conducive to disease. And so as far as some of these symptoms that you see here in the pictures, they're small brown spots with a little bit of a, well, you can see why they say like an eye. The necrotic spot surrounded by a chlorotic halo. All parts of the tree are susceptible. And again, you know, if you can keep your plants healthy, good sanitation, pruning, opening up the orchard to good, you know, management of the humidity, you can help decrease the symptoms of this disease. But I guess in recent times, what we've been seeing is an increase in incidence and severity of anthracnose and cercospora spot. And you can see that some of these lesions on the leaves are getting bigger, more widespread. And so it's interesting too that for the history of Hawaii, we say anthracnose is caused by Callitotricum species, but what species are they? So we're in the process of identifying them and characterizing them and making sure that we don't have the species that causes coffee berry disease, which I'll just mention later. Okay, I want to give the brief update that we've actually found a new leaf spotter in Hawaii, something emerging. We found some unusual spots, these left side of the picture here you're looking at. They do look circular, but the key difference between this and really cercospora spot initially was this almost like well, it looks like a little bit of tar underneath the lesion, but those are actually the spore masses produced by the fungus Paramyrothecium roridum. Now, this is well known around the world, but the first report here, and it's just showing you D is what the fungus looks like on the plate. C and E are examples of the spores that get around, again, because of wind, because of rain splashing, just general movement. And of course, when we find something new, the first thing we have to do is prove pathogenicity. That's what we were doing in those right panels, recreating what we were seeing in the field. And then unfortunately, we not only started finding it a little more in the field, but under some conditions in a greenhouse. And you can see, again, it seems to be, which is a record around the world. It causes more of a problem on small plants in a greenhouse setting, larger lesions, necrosis, and even necrotic areas of the small stems. So you can see where this really could cause a problem. Okay, when it comes to coffee leaf rust caused by the fungus Himalaya vestatrix, as everyone's well aware, it was first discovered on Maui in 2020. Within 8 months, spread throughout the state. We've been looking at various aspects since its incursion, including diagnostics and early detection, race, doing a lot of work with fungicide testing. Today, I'm really going to give a bigger summary of the variety testing and my favorite aspect, the natural enemies. And so really just to, as the reminder of Himalaya vestatrix here in Hawaii, we now have the capacity to do rapid testing, not only with PCR and sequencing, but a molecular qPCR test, kind of virtual, real time, take a sample and within essentially maybe an hour, we can tell you if it is that. So that is helpful, especially when you don't see those characteristic spores. We've been collectively doing a lot of genotyping work to figure out where it came from potentially, how it's getting here, with Dr. Ames' group at Purdue and Dr. Kokbach at ARS on the East Coast. We helped figure out what race it is. Now, there's more than 55 plus races, and working with the experts in Portugal and a set of differential plants that they inoculate with our strain to figure out that it was race 24, which means that it contains these virulence genes 2, 4, and 5. We knew already everything grown here is highly susceptible to the race we have. And it really is critical to avoid the introduction of new races, which could cause disease on a number of varieties, maybe that we're hoping to have in the future. We also need to really avoid the evolution of new races. This pathogen is I mean, kind of amazing and what it can do and evolve. If there are a lot of germplasm, so different varieties growing in the same location, some of them being resistant varieties, over time, it is possible to have the evolution of a new race to overcome that resistance. So you have to remember now, you can't just plant a resistant variety without managing in other aspects. That's going to be key. Okay, so to kind of move into, I guess, the two big areas that we're involved in, with variety testing being the first. And this was to understand as far as the resistance screening here, to try to get our hands on as much material as we could already growing here, anything from the tiniest seedling to trees in the field. The first being optimizing a way to do rapid screening. And you can see here the picture on the bottom, there are these leaf discs, and that's a great way to rapidly screen a bunch of material with minimal space required. We can now prioritize the varieties, what we need for additional testing. Initially, this stage, we're looking to see what material is susceptible. Okay, is the pathogen reproducing and producing a lot of spores on this material? Well, that won't be good for future testing. And it was really key because there was this excellent germplasm, all these varieties that Dr. Chifumi Nagai and team at HARC had done a lot of breeding in the 80s into the 90s, most not being tested because the pathogen wasn't here. How could we get through collectively as much material as possible in the shortest amount of time to see if resistance occurred? And so these were HARC sessions with F2 and F2 catamore crosses. So the resistant material, crossing it with all of the elites currently growing in Hawaii on the various islands. You can see ramping up from a single layer to multiple chambers containing multiple boxes of hundreds at a time. Okay, this is to show you kind of close in examples of these leaf disc bioassays. And A is the Conotypica, so that's our very susceptible variety. You have a resistant variety like Obata in the top right. Now, these are approximately 4 weeks after inoculation. We start with clean material. We apply some of the spores of Himalaya vistatrix. We put them under the right conditions with light and temperature and humidity, and we watch for symptom development and spore production. Okay, so the bottom left is a resistant catamore. No spots or spores. D is one from Maui. You can see highly resistant, but when you start this breeding material, E in the bottom right is now a resistant variety. And this is a kind of a close-up to show you what this pathogen really looks like and what it's doing and the results that we're seeing with our leaf disc assays. Okay, the left is a very susceptible variety, and you can see large amounts of spores. And that's so it's the leaf disc you're looking at the top, and then zoomed in even more under the dissecting scope to see those typical very yellow, powdery looking spores produced on the leaf. Now, we've been seeing, in a way, two things that we're calling resistant. The middle kind of being more tolerant, because what happens is the pathogen, once it's inoculated, it does enter into the stomates. It starts its infection. Um, so you see a bit of these chlorotic spots, which are the first symptoms of disease of coffee leaf rust, but there's no spore production. So these varieties, in a way, keep the population at a level where the pathogen doesn't complete its life cycle. And with no spores being produced, you're not going to have those next rounds of diseases. Okay, so that's important. We call it tolerant or resistant. And you do have to remember, we actually inoculate at a very high concentration. So in a way, it's like probably higher than what typically would be seen in the field. So this is even, better news as far as small chlorotic spots, but no spores. And then on the very right, there's the example of, even though it has been inoculated, you see absolutely nothing, no symptoms, no spore development. Okay, so the team really went, they were amazing and turned into a machine. And so here's the first essentially year or so just to show you what we're looking at. These are all the various successions. And I should mention the team, awesome, from HARC, we developed a way to get leaf material from Oahu sent to our lab here so we could set up all these leaf discs and test in PBARC well within, the chambers that we have here, which was very important, especially the early stages of disease on Hawaii Island. It was well contained. On the x-axis, again, you can see a variety of the accessions. And then on the y-axis, it's the percentage of the discs that's sporulated. And so with these populations, you can see from the very left, there are discs with none. No 0% sporulation. And then when you're looking at approximately 16 discs per plate that I showed you, when you see some of the controls to the right, essentially everything's sporulating. And now you see this kind of curve here. When you keep going, as of January of last year, you can no longer read anything. I don't expect you to, but you could just see that it's a common trend. Lots of zero sporulation, and then some of them are still susceptible. What's very interesting is some of them maybe have no spots as well. So this is the culmination as of hot off the press of this month. We have tested, and I say the team, amazing team, has tested close to 800 cultivars, and happy to report that 76% of them are resistant to coffee leaf rust. And again, just to remind you, that very susceptible picture in the top left, tons of spores. You have this intermediate resistance where with our testing at a heavy inoculum load, you do see spots, little chlorotic spots, but you actually don't see spores being produced, to many, many highly resistant, seeing no symptomology and no spore production. Okay, this is another way to look at it. This is actually the average sporulation percent, and how many of those accessions show it. And the best thing you can look at is the very left. So of close to 600 accessions having no sporulation whatsoever. Okay. So that was one huge population. The team collectively and growers, people in the field that would see trees. Here's an example, a heavily infected field where a couple trees had a full canopy, even though coffee leaf rust, really the severity was very, very high in this field. So the material we would take the leaves, actually do these leaf disc assays, show you the comparison of the very susceptible the typica or the bourbon on the bottom right. Again, you put the spores on, within about 3 to 4 weeks, you're seeing the necrotic spots and spore production, and these other trees being highly tolerant or resistant. And this is one example where it went into this pipeline. It was named Maite. So not only was it initiated into tissue culture, it was sent to HARC to further multiply it. It's been genotyped by Dr. Dapeng Zhang, ARS in Beltsville, and it tastes good. So this is great news, something already here moving forward. Okay, we also worked very closely with Andrea and Matt and their excellent trees growing at the Kona Research Station helped test all of the Catamore crosses. And we are now complete. We've tested 110 of the trees, and almost 93% show this tolerant or resistance. When you're looking, so no spots at all, 41 of the 110, 62 being spots but no spores, again, which is great news because the pathogen is not completing its life cycle. Again, it's a high concentration that we put on, and very few showed CLR. So this does follow with what Andrea and Matt have been seeing in the field and multiple seasons, that backs up with these leaf disc assays. And here's the summary. So it's really great. This is a great assay, a great first look to prioritize, to confirm, when trees are showing good survivability in the field. That's what we're seeing when we're testing. Okay, I'm going to go really quickly. I know that was a lot. But like I say, my other favorite project, we're looking for biological control of coffee leaf rust. In our case, they're fungi attacking fungi out in the field. It shows a little picture in the top right. Those are the spores of Himalaya, and you see this white fungus attacking these spores. Well, we did a huge survey for the first time ever in Hawaii. We see all this diversity of these fungi. We purify them, identify them, trying to characterize them, and see what are they doing and can they be used as a biological control. And we can use these leaf disc assays again to see if there's some promise and motivation for moving forward with additional research. Okay, we kind of look at it two ways. To look at its efficacy, we can see, well, we found it colonizing spores. Now, when we treat spores from the field, we make leaf discs having these spores, and we apply each isolate of these potential mycoparasites. What do they do? Do they colonize? Do they use it as a food source? And take over the population. And you can see the graph. We're trying to figure out just the biology behind it, how well it grows and colonizes. And so definitely spraying some of these isolates, oh, they like the CLR spores as a food source. Okay. We also can look at it as a protection, a way of, some of the other fungicides that are suggested for the field, protecting leaves before infection occurs. And that's what this assay is, that if we spray these mycoparasites, again, which are already in our environment, onto leaves, and now you inoculate these leaves with coffee leaf rust spores, the Himalaya, 24 hours later, what happens? Okay, so we would do that. We would watch and wait symptom and sign development over time, looking to see if there's those chlorotic spots, those spore production, and how severe disease occurs. Okay, these I'm just going to end on a couple which we think are super exciting pictures. The top left being the control. Okay, so in this case, we just used a little detergent solution where we spray the leaf and then we inoculate, and it's a negative control. So we expect to see disease. We want to make sure the assay is working correctly and we do get spore production. And in this case, we used Preaxor as a control just to make sure that, okay, we know it's effective and it stops all symptoms and signs from progressing. And here's an example of three different isolates that essentially are preventing Himalaya from completing its life cycle. You do not see any spore production. So we're thrilled with some of these results. So that as far as a protectant, but it's also, we're seeing the biology of how these things are working, these fungi, that not only are they there and present, and in some cases, these isolates actually are in a way protecting the leaf. It's very difficult. Maybe it's stopping the germination, decreasing the germination, producing some kind of secondary metabolites that's inhibitory for Himalaya to complete its life cycle. But they're also colonizing the leaves. So if and when these are different, some of the different isolates, they will continue to attack the spores. So now that even though the spores of Himalaya are being produced, these fungi are now, oh, they see their food source, they're going after it. So again, these are great pictures to see. And indicating we have potential of things to move forward with. Now, it's still a long road ahead. We want to continue this. We want to understand what they're doing, when they have to be applied. We've just started to move into potted plant assays. Okay, the next step. We're hoping to then get into the field again to test a very small scale. But these are things we've already collected from the field. We want to see what they're doing, how they're doing it, and how they could be used potentially as a valuable tool as part of an IPM strategy for the farmers. Okay, I'm just going to end by mentioning things we do not have here in Hawaii, and I always want everybody to keep a lookout. I mentioned the Calototricum species causing coffee berry disease, which it is not known to occur outside of Africa, okay? But this Kahawai species, that's why we need to understand what we have here. The critical thing with this species is that it will affect, actually reproduce, cause more spores on green berries, which is vastly different than anything that we have here. Coffee leaf scorch is caused by Xylella. So if you see this either witch's broom, this very small canopy type, that could be indicative of this well, it's kind of a bacteria-ish that can't be cultured, a whole different uh, scenario. And also just pay attention to a very rapid death, coffee wilt caused by fusarium that we do not currently have here. The key symptomology, seeing those dark veins on the leaf material. Okay. If you do, please reach out to Andrea, Matt, myself, just to either rule it out or to catch something potentially early. And on that, I will say thank you very much. I appreciate your time. I'll try to answer any questions. And if not, please feel free to reach out. There's my email, lisa.keith@usda.gov. Thank you very much.